All right, so I'd like to thank the entire Cancer Symposium for maybe making this event possible. I really enjoyed this event, and thank you for the opportunity to help me present this. So I'm here to talk about DriveRank, which is a method that we developed in order to detect patient-specific driver mutations in cancer. And I'd like to open by starting about describing the mystery of driver mutations in a computational sense. So over the last few years, recent advances in DNA sequencing technology have greatly enhanced our ability to analyze cancer genomes because next generation sequencing technology has allowed us to detect a large spectrum of somatic mutations in cancer genomes. And with the more complete genome set, we are able to find more and more mutations in cancer, which could lead to a selective advantage over tumor cells. And knowing these mutations would greatly enhance our ability to find the source and find possible treatment methods in cancer. So these mutations called driver mutations are mutations that we are looking for. However, it is important to note that not every mutation in a cancer genome is going to be a driver mutation. In fact, most mutations actually provide no selective advantage. These mutations are called passenger mutations, and thus the ability to sift out the driver mutations from the passenger mutations has been a very big mystery in computational biology today. So there have been many approaches in order to tackle this problem, and there have been many previous methods have been developed. Most of these previous methods operate under the idea of recurrence, and recurrence is basically to say that if you're given any patient, any group of patients, whatever mutation occurs with a high frequency in those mutations are going to be driver mutations. There have been more sophisticated models using recurrence by adding another term called exclusivity, in which a subgroup of patients would include both a subgroup of patients would have a driver mutation if both that driver mutation if that driver mutation was recurrent in that subgroup and exclusively recurrent in that subgroups. And this was MIMO developed by Cyril et al. And Vanden et al.'s Dendrix program use both recurrence and exclusivity. And these software programs have been able to find many types of common driver mutations. However, these methods are limited because they are not necessarily patient-specific. And the problem with not being patient-specific is the fact that every patient has a different tumor. There are no two tumors that are alike, and it is very possible, because not all tumors are homogeneous, that there could be multiple driver mutations for different types of tumors. So thus, these methods might not provide the most direct answer in order to find a patient-specific driver mutation. So our goal is to address these limitations in driver mutation software by directly assessing the impact of mutations in each individual patient. And we decided to take a different approach than other methods have been looking for. Instead of looking for a patient population or subpopulation, what we decided to do is we decided to take a look at every individual patient and try to find out what genes what mutations in that patient are most likely to be driver mutations. And we do this by handling this like a mystery, in a way like how Sherlock Holmes would solve a case. And so we divide this into suspects and clues. Suspects we are going to limit to mutated genes, so only driver mutations that actually affect the gene will be considered. And the clues or characteristics of a driver mutation, we decided to focus on two of them specifically. One of them is interaction. How connected are mutated genes? Would a suspect mutated gene, in theory, would connect to many and would have a high impact and thus connect to many other genes in a network of genes. And also the other clue we would like to look at is expression, differential expression to be specific. And expression is to say that if a suspect mutation would be highly differentially expressed in cancer and also propagate other highly differentially expressed genes downstream. So by looking at interaction and expression, we will be able to find a method to rank individually what genes are most likely to be in cancer, be a driver mutation. So our method is a variant of Google PageRank, and also Morrison et al. a variation of Google PageRank called GeneRank, which ranks, and Google PageRank, the idea behind Google PageRank was it's the heart of Google's search engine, which was developed by Larry Page and Sergey Brin by using, and it used a stochastic random walk to determine the impact of websites. And the stochastic random walk, for those of you who are not familiar with that, it's a relatively it can be an intuitive process. My old statistics professor used to tell me that just imagine a network, for example, a network of bars on Green Street, for example, over down the corner. And if a random college student got into a random bar and spent some time there, had a little bit too much to drink, and lost a sense of direction, and just stumbled randomly into a bar next door, and then repeated this process over time until he could no longer do that, we could be able to find out the popularity of each bar by simulating this over thousands and thousands of college students. For example, like on a Friday night, we could simulate this, and thus we would be able to determine the impact of every bar. And the general theory is that a bar's popularity will be based off by the popularity of the bars that link to it. So by using this idea, we developed a per-patient ranking system that used this concept as well as factoring in differential expression genes, differential expression for the patient. So by using a differential expression by exome, from exome and RNA-seq data coupled with the network pathway, we are able to detect patient-specific driver mutations. So this is the iterative, on the top is the iterative form of our equation, and the bottom is the matrix form of our equation. And it's a little bit complicated to describe, but in the most abstract form I can say is like, it requires several terms. One is differential expression. So this is a differential expression weight here, and a connectivity form, which is a matrix or iterative representation of how well a gene connects to other downstream genes. For example, I would have 3 connections here, which would be positive. A damping factor, which is a parameter that we trained. It's basically the weight between expression and connectivity that we have. This was trained using previous data sets. And we also, because it's a stochastic random walk, and we use the rank of the outlinks. So basically, downstream genes, the rank of the downstream gene will affect the rank of the gene itself. So thus, so it's a little bit different than Google PageRank, which was rank the inlinks. However, the ideas are relatively the same. So it is important to note from this equation that every gene in a possible network will be included. However, we will only look at genes that were mutated as a comparison tool. So to validate our data, what we decided to do is we decided to take a look, compare our data with MIMO, which has been a very popular standard these days. And from MIMO's data, so MIMO detected 12 potential driver mutated genes in glioblastoma, and we did this over glioblastoma, ovarian cancer, and breast cancer from TCGA. And from this chart over here, we ranked all the genes that MIMO detected, and we found that. And we, for each patient, and we took the median of that to just so we could get a nice statistic over here. And we found that the majority of the genes in blue are ranked in the 90th percentile or higher in DriveRank, and also like all but 2 are actually ranked in the 80th percentile and higher. So we have a relatively high concordancy with MIMO. So after validating our data, we decided to take a look at a frequency analysis. So we decided to look up how many times a gene was mutated, how frequently it was significantly mutated with our results. So we took 92 glioblastoma patients, and every time a gene was mutated, we added that as a total hit. And every time it was significantly mutated, we would add that, or mutated it within the 90th percentile, it would be considered a significant mutation. So by looking at this graph over here, we can roughly classify these driver mutations as 3 types they're in 3 categories. One is common driver mutations that are relatively commonly known throughout, and there's a consensus in literature about that. Other commonly cancer-related genes, these are oncogenes that have a very high potential to become driver mutations as well. And the third group is the most interesting, actually. These are less commonly heard in literature, but that does not necessarily mean they're not driver mutations yet. So we wanted to take an in-depth look at both the common and less common driver mutations. So our sanity check over here was TP53, and it's the probably most well-known driver mutation, as most people here would have had some course that talked about TP53 in cancer. And TP53 was mutated in our data set about 51% of the time. But every time it was mutated, we detected it as significant, which means that, which is concordant to how other data sets found, by using recurrence A, also found the same, that TP53 was mutated almost every time. And the reason why it's mutated, as you can see in the graph on the right, the center TP53 connects to about 200 different genes, so it's extremely highly connective. And the genes in green have drive ranks of at least in the top quartiles, the top 25%. And you can see that there's a disproportionate amount of green nodes here. So thus, we can see that, like, because it's both highly connected and other genes are highly ranked that connect from TP53, TP53 has a very high rank itself. So furin is the gene that we wanted to look for, which is less commonly concordant with literature. And the reason why we wanted to look at furin, because it has a biological significance in cancer, because it has a lot of, it leads to protein convertases, such as the growth factor, that could lead to a selective advantage. As you can see here, PCSK and Notch in this subnetwork right here is links to furin quite well. And furin appears relatively high in glioblastoma patients 28% of the time, but it's only significant about 69% of the time. And this is probably the reason why it wasn't detected by recurrency methods, because it wasn't significant enough times. However, because we only use individual patients to do this analysis, this could be an example of a patient-specific driver mutation. And previous literature from Coppola and neoplasia actually stated that driver mutations actually inhibiting furin actually decreases the selective advantage of tumor cells. And so in summary, DriveRank is a patient-specific method to discover new driver mutations. However, and it can both find the common driver mutations from a per-patient sample with very high frequency, but it can also, as an advantage to other previous methods, identify rare mutations that are potential drivers on a per-patient basis. So if you were a cancer patient with a very rare cancer that no one else has actually done, recurrency methods would fail in order to detect your driver mutations, but we would be able to find at least a search engine-like tool to figure it out. So we would like to improve the model by improving the damping factor using a Bayesian analysis, actually, instead of having just a constant term. And also, we wanted to modify not just mutated genes, but also like non-coding regions and other changes as well, other genomic alterations. So I would like to conclude by acknowledging Pratik Lahiri, who is my partner in this project, as well as Ryan Cunningham, who helped us develop the DriveRank algorithm, as well as my advisor, Dr. Jian and the entire Ma lab, and also our funding sources, the Illinois Distinguished Fellowship, the National Science Foundation, and the NIH. So thank you very much.
All right. Do we have any questions?
I have one practical question for treating patients
Okay.
for a basis. Because it's nice what you've done in correlated expression with the disease, but how expensive do you think that would be for determining per patient treatment? And at the end of the day, how do you determine, even if you determine some genetic alteration, is that going to be determined? Is it going to be a driver? How do you determine?
Well, okay, the thing is that if right now we're limited by the cost of being able to sequence each patient's genome, we need exome or RNA-seq data for any patient that we want to categorize. So thus, I guess if we were a business and we wanted to charge for this, I guess our prices would be dependent on how much a next generation sequencing technology would be. But once we get the data, and we will be able to have at least it would be an interesting tool for physicians, I think, because once we have the data, we have a list of driver mutations that we suspect. So thus, if a physician looks at this and a physician has the biological knowledge to interpret the data, he or she would be able to provide the best treatment, because by pinpointing the specific genes that were mutated with that we found, actually. So.
All right, I think we'll, one more.
Do you think that the driver mutations could change over time as the tumor and the body sort of co-evolve? So require maybe monitoring in specific intervals?
That is actually an interesting question, because a lot of one interesting discussion I've had before was the fact that if for example one gene was mutated and that gene was for example involved in a repair pathway, especially a DNA repair pathway, that gene could actually propagate other other mutations. So yes, in theory, if we had a time factor, we currently are only using TCGA data, which is not time dependent at the moment. So, but if we did monitor this over time, we could actually find more information, and we could actually make a stochastic process within a stochastic process, our original stochastic process, and thus and actually be able to model it that way. However, unless we have like the time, but like as the earlier question just said, that it would probably require a lot more, that would actually increase the cost a lot more. And currently, like next generation sequencing technology, although it is relatively cheap to sequence a genome, it's still not into like that $1,000 range every day, you know, so you can't just go to Walmart and get your genome sequenced.
Okay, so
One question. So if I understand your method correctly, you started off with your seed genes in your network with differentially expressed genes, and then you use your DriveRank, which uses a random walk or Drummond's walk algorithm too.
Yeah.
And then you do. Then after a number of certain hits, there's probably a cutoff in significance that you choose as driver genes. My question is, if you use differentially expressed genes as your driver genes, I mean, what is your justification for using those genes as your drivers? Because those may be differentially expressed. Those actually may be passenger genes to something else at a higher level, which could be controlled. So I'm not sure how you're I'm sure if you have, I don't know what your false discovery rate or your p-value cutoff is, but you probably have many differentially expressed genes. Among the many, how are you tuning down what to choose as driver genes from a not a statistical standpoint as you use, but a mechanistic standpoint inside the network?
Well,
the second question is, what is your PPI database that you used?
Okay, so the second question first. We used TCGA data. We used three types of cancers: glioblastoma, ovarian cancer, and breast cancer. And so as actually what our significant cutoffs was actually the fact that first of all, since we are modeling driver mutations, not differentially expressed genes, what we did is actually the first cutoff before we even used differentially expressed genes, we actually took out we only looked at genes that were mutated first. A driver mutation must be a mutation, if anything. So thus, and that actually mutations only, and with mutations, we are able to significantly cut off our gene network to only about 35 or so genes that we can find. But for our network, we rank every gene independently, whether or not it's mutated or not. However, we only I guess care about the genes that are actually mutated. And thus, for any particular patient, the data sets that we found from TCGA was actually, I guess the TCGA data came from UNC as well as Broad and Washington University and Baylor, and a combination of those. And the mutation data was only mutated genes, only a couple of them were actually significant. We used about 10% cutoff. So that actually made a very manageable number of 5 or 6 per patient. So going through that, we would be able to find, we would actually have a very manageable number of genes that we suspect to be driver mutations. And a lot of them like P53 and EGFR are all very commonly documented as driver mutations anyway. So the problem is we have no goal. The problem, I guess, with the analysis itself is the fact that we don't really have like a good false discovery rate because we can't but no one can because there's no like gold standard in defining a driver mutation. There's no consensus about like what is a driver mutation and what is not a driver mutation, and that's actually a very big problem. And that's why we develop algorithms like this in order to find out which mutations are the driver mutations. So.
All right. Well, thanks a lot.